Rick Russell.
Thanks, Ricky, and I'd like to thank the organizers very much for putting all this together. Before I talk about research from our lab, I want to say a few things about Dan and Dan's lab and my experience from being a postdoc in Dan's lab. So in the fall of 1996, as I was finishing my graduate work, I did what was really a fairly systematic search for postdoc labs with, at that time, an interest in enzymology. Dan's lab hit on several counts. Enzymology, obviously. I had a vague, undefined interest in RNA at that point, and of course, Stanford was a plus. But the thing that really defined my choice was reading the papers that were coming out of Dan's lab at that time. And I remember these three particularly. It just struck me that they were so insightful, rigorous, and complete, so much more than anything else I was reading. And I remember thinking, I want to learn to do that. I was lucky. Dan was agreeable to that plan. So I was a postdoc from 1997 to 2002. And just to put this time frame into reference, the view from the Beckman Center looked something like this. And Dan looked something like this. We can discuss which one has changed more. But what I want to do is share some things that I learned from Dan. And there are two disclaimers here. One, this isn't an exhaustive list. And two, some of these we've heard in various formulations before this morning, but I'm going to say them again because these are benchmarks that I've tried very consciously to hold myself accountable to in my career and to communicate to students. So first, make sure you're asking important questions. I think anybody that's been in Dan's lab or has interacted with him extensively has heard some version of, that's fine, but I have no idea from anything that you're saying why I should care about you. Second, equally important, make physical models that are consistent with all of the data, not just your cherry-picked favorite data, and at least as important, that make specific predictions. And then to test those predictions, particularly the ones that are orthogonal in some way, orthogonal conceptually or in terms of methodology. And then last, be brutally honest with the data, with yourself about the data, skeptical of any model, particularly your favorite model. And as I've progressed through my career, I've really been kind of surprised to see how many scientists there are, even established and acclaimed ones, that really just don't do this. So sorry. The most important thing I want to say is I'm so glad to have the opportunity to be here and say thank you, Dan, and to celebrate with all of you Dan's contributions to science, and for many of us to our development as scientists, as educators, and as mentors. And so with that, I'll take questions now. So in the rest of my time, I want to tell you about some recent work from my lab. So we've, over the years, been focused on RNA folding and helicase proteins that function as RNA chaperones, as well as additional work on RNA folding. I've had the good fortune for some of that to be involved in continued collaborations with Dan. More recently, we've also looked at a helicase that can disrupt quadruplex structures. But what I want to tell you about today is something that's really completely new to the lab, and that is a CRISPR-Cas enzyme called Cas12a. So since their discovery about a decade ago as bacterial defense mechanisms, it's become abundantly clear that CRISPR-Cas enzymes have enormous potential in gene editing applications. So they're already used extensively in research to generate knockouts in mice and other model organisms. They're beginning to see widespread use in the food that we eat, and one can readily imagine a time where they're centrally involved in medicine, potentially curing single gene diseases like sickle cell anemia, and even playing important roles in combating complex diseases like cancer. For the future use and development of CRISPR-Cas enzymes, it's a key challenge to understand the molecular mechanisms by which these enzymes target DNA sites and cleave those sites. We're also interested in this question from a fundamental scientific perspective, as these are interesting reactions biochemically and biophysically. So CRISPR-Cas enzyme binds its DNA target in 2 steps. It first forms contacts with the protein to a conserved sequence known as the PAM sequence, and then the guide RNA with the enzyme forms specific base pairs to the target. The formation of this R loop licenses nucleic acid cleavage activity, which results in a double-stranded break in the DNA that typically following DNA repair processes leads to a change in the sequence that can inactivate the gene. A key issue for gene editing applications is the extent to which the enzymes are able to discriminate against a perfectly matched target from one that differs at one or more positions, and these are generally referred to as off-target effects or recognition. This is a critical issue for editing applications because obviously it's very important to generate breaks at the site you want to make them at and not at sites in the genome that resemble that sequence. So we focused on CRISPR-Cas12a, which has received considerable attention recently as an alternative to the sort of gold standard Cas9 for these types of applications. Cas12a is simple like Cas9 with a single polypeptide. It's also simpler in some ways than even in Cas9 because it uses a single guide RNA and it processes itself. It binds to a PAM sequence that's a fairly simple sequence and then forms the R loop between the RNA and the target, which then allows what's thought to be a single nuclease site to cleave both strands of the target, generating a staggered end. Really most importantly, Cas12a has received a lot of attention because it's been reported in cell-based studies to be more specific against off-target effects than Cas9. However, these are principally in vivo experiments, and the observed differences might arise from differences in DNA repair or even differences in cell type between the experiments. So what we wanted to do was look very specifically at the enzyme itself, Cas12a, and measure specificity. So I know this sort of thing is familiar to everyone in the room. We decided to generate a kinetic framework, specifically to measure the rate constants of binding, dissociation, and cleavage of both strands, with the idea that this framework would be foundational for assessing the effects of mismatches. So we used purified Cas12a and a synthetic guide RNA and DNA targets consisting of 2 oligonucleotides. We measured the rate constant for binding by mixing Cas12a with radiolabeled DNA, adding unlabeled DNA as a chase, and then separating bound and unbound DNA on the native page. I might have just said RNA, I meant DNA. So we can see binding over time here with rate constants that depend on Cas12a concentration to give a second order rate constant of 10 to the 8th per molar per second within 100-fold of the diffusion rate, and a plateau here that additional experiments have shown represents the rate constant for R loop formation at about 0.1%. An analogous method was used to measure the dissociation rate constant, and we found that dissociation from the matched target, and this is with a mutant of Cas12a that's deficient in nuclease activity, so that it will dissociate. This is extremely slow on the time scale of hours to days with a rate constant of 6 times 10 to the 6th minus 6 per second. So what I've shown you so far is binding is really fast, dissociation is really slow. This leads to a calculated value for the KD of 50 femtomolar, which is as much as 6 orders of magnitude tighter than previous reports in the literature for this enzyme, but is actually in the range that one would expect for formation of a 20 base pair R loop. We also measured the kinetics of strand cleavage, and there's some interesting other stories there, but in the interest of time, I'll skip those data and just make the simple point that the rate constant for DNA cleavage is many orders of magnitude faster than dissociation, indicating that essentially every binding event results in DNA cleavage, or in other words, that binding is rate limiting, and cleavage specificity is determined by differences in binding kinetics, not by equilibrium. Again, I don't probably need to show this figure for this audience, but the specificity determining step can be understood in this free energy profile. But other audiences, they really don't get it until so cleavage is much faster than dissociation, and if there were a change in the sequence that destabilized the complex by accelerating dissociation, this wouldn't lead to enhanced specificity because you're not changing the highest rate. Okay, so this meant that we were really interested in looking at the effects of mismatches on the binding kinetics. Let me be very clear what I mean by mismatch here. At each position, we switched the positions of the purine and the pyrimidine like so. So in all cases, the DNA forms a base pair to itself, but by switching these, we've created a mismatch between the target strand of the DNA and the guide RNA. Okay, and then we use the same methods to measure the binding kinetics for each of these mismatched DNA targets. And what we saw was that there are substantial effects of the mismatches slowing down the binding kinetics by 5 to 1,000-fold. So here, the rate constant for binding for each sequence change is shown relative to the wild type, and a bigger bar is slower binding. We were particularly interested to see that the effects of these mismatches extended away from the PAM sequence. So here we're starting at the PAM sequence and going away. Yeah, there are large effects throughout the R loop, even at PAM distal positions. This is interesting because it's quite different from the basic properties of helix formation in solution. So in solution, as we know from the work of Dan, Steve Block, and others, helix formation is nucleated in just a few base pairs, and then the rest of the helix rapidly zips up in a downhill process, so the specificity would be determined in only these first few base pairs. Our results suggest that Cas12a has shifted this transition state later, or in other words, made the early part of the R loop propagation more reversible until eventually this transition state is reached later in the process. So we were especially interested to then compare the properties of Cas12a with Cas9 directly to see whether this was really a difference. After all, Cas12a and Cas9 use identical 20 base pair R loops, so the simplest expectation might be that the properties would be the same. However, we found that that was not the case, and instead, consistent with previous work of others on Cas9, there's strong discrimination at the PAM proximal positions, but then this falls off, and in the limited number we've tested, there's really very minimal specificity. These are just 2 to 3-fold specificity against these mismatches. So it appears that Cas12a really discriminates more strongly than Cas9 against PAM distal mismatches. And we also measured the kinetics of dissociation for each of these mismatched positions. And here again, the results are shown relative to the wild type, where a larger bar is faster dissociation for the mismatch. Consistent with a late transition state for binding, we see larger effects of the PAM distal positions, as one might expect. Then if we combine the effects on binding and dissociation, we can see the effects on equilibrium for each mismatch. So here we're just adding the heights of the bars. And the striking thing here is that there are fairly large 100 to 1,000-fold effects that are fairly constant throughout the R loop. At least there's no trend in a direction. Sorry, this is particularly interesting because Cas12a and Cas9 are described as having a seed region in the PAM proximal positions, one definition of which is enhanced thermodynamic specificity against mismatches, so we don't see that here. You can see that there is variability between these positions, but the striking thing is that both the magnitudes of the effects on equilibrium and a lot of this variability can be accounted for simply by nearest neighbor predictions for these R loops. And so what's shown here is a comparison of the measured values and the calculated values for the extent of destabilization by each mismatch. And you can see there's a general correspondence, even in some of the differences. That correspondence very definitely falls off here at the end, where the mismatches are mattering much less than they would be expected at the most pan-distal positions. And we don't understand that effect.
Rick, I'm going to interrupt you because where's 14? That was the one that was off before. From 13 to 15. You know, like a lot of buildings don't have a 13th floor.
That's well.
Okay.
Thank you, Dan. I don't know. I'd say that's Isabel's fault, but that comes back to accountability. All right, so let me just conclude by hitting the main points of what our data shows. So, Cas12a binds very tightly, rapidly and tightly to a matched target, and then cleaves that target much faster than it dissociates. So binding is rate limiting. However, the late transition state in R loop formation generates high specificity despite this rate limiting binding. So here, imagine the R loop forming in these groups of several base pairs with this late transition state. So if there's a mismatch that impedes R loop formation, increasing the barrier here and generating specificity. I didn't show the data, but this is the only step at which there's a difference for mismatches. So the cleavage rate constants are identical after the R loop forms. And then the last point, and most general with this high specificity, Cas12a has promise for gene editing application. So I'd just like to finish by thanking the people responsible for the work. This is all the work of a very talented graduate student in the lab, Isabel Strokendahl. It's also a close collaboration with Ilya Finkelstein, my colleague at UT Austin, and Jim and Fatima in his lab. I'd like to thank these funding sources. And last, coming back to Dan, I'd like to thank Dan and Namita and Inga for publishing what was part of the inspiration for the work, a thought piece on specificity in CRISPR-Cas12a. And thanks for your attention.
Time for a couple questions.
That was a great talk. I was wondering, can you explain these kinetics as the rate limiting step being actually the melting of the DNA strand that it's invading? Would that account for why the rules basically predict the stability of the D form?
Right, right. I mean, the answer is I don't know. Models you can imagine are that it's more reversible than helix formation because you're kicking out that other strand of the DNA. But it can't be just an intrinsic property of that biochemical process, or it would be true in Cas9 too. So maybe there's something about the enzyme that makes it more reversible. I'm sort of dividing this into two groups. It's possible that it's more reversible at a base pair by base pair level, or another possibility that would also account for the data would be if there's like a checkpoint at some point toward the end. Right? It doesn't have to be reversible base by base if it goes a certain and it's all reversible, all the way from there. And we're interested in that and working on it.
So the subsequent molecule work from the Dogma lab that the Cas9 enzyme will go through conformation changes and low, medium, high phase states, and somehow the distribution of these states are sensitive to base mismatches. So how does that information map onto the model?
So there's less work done for Cas12a than Cas9 for that type of thing, but there clearly are conformational changes. That's made apparent by some different crystal structures, and also the way the enzyme works. The formation of that R loop activates the protein for DNA cleavage. For Cas12a, and this is also work from the down group, it turns out that it'll also, with high efficiency cleave single-strand DNA is added in trans. So upon formation of the R loop, there is apparently a conformational change that makes that nuclease site highly active. whether that, and it's possible that that conformational change also has to do with this reversibility of Farley formation, pursuing that further.
Anything known about, so there are two chemical steps, anything known about that, or are they inaccessible? In other words, are they, go in the same order, is there one that's favored over the other? How do you get both of them into the active side?
Right. So cleavage of the non-target strand is about 10 times faster than cleavage of the target strand. The non-target strand is the one that's displaced and is sort of over near the active site. There's evidence that there's a single active site. We don't know whether that's an obligate order or it's just random, but that strand is closer.
So it's an experiment you can do by presumably putting a phosphatidyl.
Yeah.
Any other questions for Rick as Katrina said? No?
Yeah.